We figured out vault structure, identified all the vault components, and we showed how cells assemble the particle. However, we were unable to figure out vault function. Whenever I gave a research talk, I would always be asked the same first question. Professor Rome, what do vaults do? I was getting frustrated by the question, so I would usually go for a laugh and say, the function of vaults is to put my 3 kids through college. Eventually, I consulted with my wise uncle, John Fitzgerald Rome, who shared his sage advice. Ask not what vaults do, ask what you can do with vaults. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. With the expert assistance of my executive producer, Tara, I'm trying to tell the vault story and teach some cell biology to anyone who is interested. In my last video, I showed you the amazing process of vault assembly. If you haven't seen it, I'll put a link below. Today, I want to cover 3 things. First, I want to tell you about our struggles to get vault research funded. And second, how we developed a new interdisciplinary project called a NIRT grant. Finally, I will describe our first attempts to engineer the vault. Stick around and I'll tell you how we turned vaults into nano-light bulbs. I wish I could say that the idea for engineering the vault came to me during a moment of divine enlightenment. Unfortunately, that's not how it worked. It was late in the 1990s and we were struggling to keep vault research funded by the NIH. As I mentioned in episode 5, my first vault grant to the NIH got the best score I had ever gotten. Our second grant was initially rejected and only funded after 2 tries. The NIH gives a grant only 3 tries, and our third grant took all 3 attempts before we were finally funded. When our third grant ended, we wrote a new grant, and again, we tried 3 times to get it funded by NIH, but unfortunately, we were rejected. It seemed like my lab was the only group that thought that vaults might be important and that understanding vault function was a valuable scientific undertaking. NIH didn't care that I thought vaults were cool. It reminded me of this cartoon where 2 guys in white lab coats were talking, and one guy says, "I asked Santa for a research grant." The other replied, "You still believe in research grants?" Faced with the dire research funding situation we were in, we decided to embark on a journey to engineer the vault particle. And by engineering the particle, we could give the vault some new functions. This was just about the time that we figured out how to make empty vault particles by expressing the MVP gene in insect cells. I told you about this in episode 10. The National Science Foundation, NSF, is the 2nd largest funder of academic research grants in the U.S., but they don't usually fund grants that are medically related. I heard about an NSF grant called a NIRT grant. NIRT stood for Nanoscale Interdisciplinary Research Team. I had the idea that maybe NSF would be interested in a grant that proposed using vaults as a new type of nanotechnology, but I knew that I couldn't say a word about the medical significance of studying vaults and engineering them. To be clear, I did want to eventually use vaults therapeutically, perhaps as drug delivery vehicles. However, we would need to understand the properties of vaults to get started. NIRT grants are structured from interdisciplinary teams of academic researchers. So I needed to put together a team that would study vaults as nanoscale particles. My dream team was a bunch of superstar nerds funded by a NIRT. UCLA is a terrific place to do collaborative research, so I went digging online through bios of my UCLA colleagues in the schools of engineering and the life and physical sciences. I found a handful of scientists who I thought might be interested in studying vaults. I called each of them on the phone and I said, "You don't know me, but I have this particle that I think you might be interested in studying. Could I come over to your office and show you a few pictures?" Everyone I called was open to hearing my story. Each one, after hearing my vault pitch, was interested in joining a vault collaboration. My top nerds included Halmon Bouquet, Bruce Dunn, Sarah Tolbert, and Jeff Zink. We started meeting together, tossing around ideas for engineering the vault particle, and within a year we had submitted and were successful at getting a NIRT grant. This grant enabled us to try lots of interesting applications for the vault particle. One of the first things we did was figure out how to package foreign proteins into the vault. In episode 8, I described how Valerie had cloned the vault protein VPARP. Her research revealed a fragment of VPARP that was still able to tightly bind to MVP. This piece of VPARP turned out to be about 10% of the VPARP protein. This domain, which we called the MVP interaction domain, or INT for short, binds to MVP at this location and is responsible for VPARP binding to the inside of the vault. In other words, this domain acts like a zip code to direct the protein into the particle. We reasoned that if the INT sequence directs VPARP into the particle, then perhaps it could direct other proteins to go inside. So, to prove that INT could serve as a zip code, we collaborated with the Zink, Dunn, and Stewart labs to engineer a protein that was originally isolated from fireflies. It's called luciferase. Luciferase is an enzyme that gives the firefly its glow. Because of this property, the luciferase reaction is very easy to detect, and it's widely used by researchers as a valuable tool for studying biology. Here's a simplified explanation of what is happening. Luciferase binds to a small chemical called luciferin. A chemical reaction called oxidation occurs. The luciferin is converted to oxyluciferin, and the reaction produces a kind of light called bioluminescence. We used a routine molecular biology approach to attach the VPARP zip code INT onto one end of luciferase. The protein we produced is called a fusion protein because it's a fusion of luciferase and the zip code INT. When we expressed the luciferase fusion protein and MVP together and examined the vaults that formed by cryo-EM, they looked normal from the outside, but inside we found the luciferase protein packaged into two rings inside the vault particle. In fact, the luciferase inside the vault retained its enzymatic activity, meaning when we mixed these vaults with the other chemicals needed for the bioluminescence reaction, the vaults could produce the yellow glow. So we had succeeded in making little nano-light bulbs. I'm Lenny Rome, the Vault Guy. Join me in my next video where we ask the question, can the INT-tagged fusion protein get inside an intact, fully assembled vault? I think the answer will really amaze you. It surely blew my mind. Finally, if you find these videos useful or interesting and you would like to see more of the Vault story, subscribe and give us a like.